Hello students, today we will study photorespiration. This video will include what is photorespiration or Warburg effect, Rubisco enzyme, steps of photorespiration, differences between respiration and photorespiration, and significance of photorespiration. Now what is photorespiration in plants? In the 1920s, the German biochemist Otto Warburg discovered that plants consumed oxygen at a higher rate when they were illuminated with bright sunlight accompanied by high temperature. He also found that increased rate of oxygen consumption inhibited photosynthesis. So, stimulation of oxygen consumption by light is now referred to as photorespiration or Warburg effect. Let us see the definition. A respiratory process in many higher plants by which they take up oxygen in the light and give out carbon dioxide, contrary to the general pattern of photosynthesis. So respiration in light is photorespiration. You can see in this diagram, on the left hand side is the photosynthesis process. It is an anabolic process. The plant takes up carbon dioxide and light and produces sugars. Whereas in photorespiration, it is a catabolic process. The plant starts taking up oxygen and starts giving out carbon dioxide. So both are opposite processes. Now there is an enzyme called RUBP oxygenase carboxylase Rubisco enzyme, a key enzyme in photosynthesis. It is the molecular equivalent of a good friend with a bad habit. Rubisco can perform two functions, RUBP carboxylase and RUBP oxygenase. Photorespiration leads to the loss of already fixed carbon. Photorespiration wastes energy and decreases sugar synthesis. So, when Rubisco initiates this pathway, it is a harmful process. Now you can see here, Rubisco enzyme can perform two functions, carboxylation and oxygenation. That is why it is called as ribulose biphosphate carboxylase oxygenase enzyme or Rubisco enzyme. It can perform carboxylation when the temperature is low and high CO2 is to O2 ratio and CO2 is fixed in the Kelvin cycle. Whereas in oxygenation, the process occurs when the temperature is high and CO2 is to O2 ratio is low. So this process is the photorespiration. Now what is the role of Rubisco in Kelvin cycle? It is the first enzyme which catalyzes the first reaction, that is RUBP accepts carbon dioxide and forms a 6 carbon compound which is not stable, and ultimately it forms a 3 carbon compound which is a stable product, that is phosphoglyceric acid. So ribulose 1,5 biphosphate takes up CO2 in the presence of RUDP carboxylase and forms a first stable product PGA, phosphoglyceric acid. So this is the role of Rubisco in Kelvin cycle. What determines Rubisco's activity? When a plant has its stomata or leaf pores open, carbon dioxide diffuses in and oxygen and water diffuses out. Sugars remain in the plant. Photorespiration is minimized. However, when a plant closes its stomata, for instance to reduce water loss by evaporation, oxygen from photosynthesis builds up inside the leaf. Under these conditions, photorespiration increases due to the higher ratio of oxygen to carbon dioxide. So it is the ratio of oxygen and carbon dioxide and the temperature which determines the Rubisco's activity. Rubisco has high affinity for oxygen when temperature increases under light conditions. At mild temperatures, Rubisco's affinity for carbon dioxide is 80 times more. Remember, at mild temperatures, but when the temperature increases, it shifts to its oxygenase activity. So the bottom line is that dry, hot conditions tend to cause more photorespiration. Now, what are the conditions for photorespiration? Light, temperature greater than 25 degrees centigrade, oxygen high concentration, carbon dioxide low concentration. Now this is the flow sheet of the whole cycle. This photorespiration involves three cell organelles: chloroplast, peroxisome and mitochondria. Now in the chloroplast, this RUBP, instead of performing carboxylase activity, Rubisco enzyme shifts to oxygenase activity, and this RUBP takes up oxygen and forms PGA and phosphoglycolate. RUBP is 5 carbon compound and it combines with oxygen to form a 2 carbon compound and a 3 carbon compound. This phosphoglycolate is now converted to glycolate. This glycolate moves to peroxisome, where it is converted to glyoxylate and hydrogen peroxide. Hydrogen peroxide is a harmful molecule. Glyoxylate is converted to glycine. This glycine moves to mitochondria and it releases a molecule of ammonia and carbon dioxide and becomes serine. The serine is transported back to peroxisome, where it is converted to hydroxypyruvate and glycerate, which again enters chloroplast and ultimately enters the Calvin cycle. So this phosphoglycolate has to travel and take lot of time and ultimately get converted to glycerate and enter the Calvin cycle. So this process is taking time and also it is removing carbon dioxide and taking oxygen. So it is wasting of energy. Now this is the detailed mechanism of photorespiration. In chloroplast, RUDP plus oxygen in the presence of Rubisco enzyme is converted to 2-phosphoglycolic acid and PGA. 2-phosphoglycolic acid is converted to glycolic acid. Glycolic acid enters peroxisome and forms glycoxylic acid, undergoes oxidation, and H2O2 is also formed, which of course is broken down into water and oxygen by catalase enzyme. Glycoxylic acid plus glutamic acid forms glycine and alpha ketogluteric acid. Glycine moves into mitochondria, where it is decarboxylated, where it is converted to serine, and a molecule of carbon dioxide and ammonia is released. Again the serine enters peroxisome and it is converted to hydroxypyruvic acid and glycine. This hydroxypyruvic acid is later converted or reduced to glyceric acid. This glyceric acid plus ATP, glyceric acid enters chloroplast again and it is converted to phosphoglyceric acid in the presence of ATP and glyceric acid kinase. This PGA enters the Calvin cycle for synthesis of carbohydrates. You can see here the process once again. Here is the Rubisco enzyme, this is the Calvin cycle, and this is the photorespiration process, which involves three cell organelles, that is chloroplast, peroxisome and mitochondria. Photorespiration wastes energy and steals carbon. Photorespiration begins in chloroplast when Rubisco attaches oxygen to RUBP in its oxygenase reaction. Two molecules are produced, a 3 carbon compound 3-PGA and a 2 carbon compound phosphoglycolate. 3 PGA is a normal intermediate of the Calvin cycle, but phosphoglycolate cannot enter the cycle, so its 2 carbons are removed or stolen from the cycle. To recover some of the lost carbon, plants put phosphoglycolate through a series of reactions that involve transport between various organelles. 3/4 of the carbon that enters this pathway as phosphoglycolate is recovered, while 1/4 is lost as carbon dioxide. The differences between respiration and photorespiration. Respiration process is the process of energy release by the breakdown of substrate material. It is the phenomena of consumption of oxygen and release of carbon dioxide in the presence of light. Respiration takes place in dark in the plants. It takes place in the light in the plants. It takes place in cytoplasm and mitochondria. It takes place in chloroplast, mitochondria and peroxisome. What is the significance of photorespiration? It has photoreceptive effects, that is, preventing light-induced damage to the molecules involved in photosynthesis. It helps to maintain redox balance in cells and supports plant immune defenses. Also removes toxic metabolic intermediates like hydrogen peroxide. Significances are less. So we come to the conclusion that Rubisco is a very important enzyme in plants for energy production. It can perform 2 functions, that is carboxylation and oxygenation. Photorespiration, overall it is a wasteful process where sugars are not synthesized. Thank you.